Hey everybody, so I figured that given our last video we talked about polypores, I figured that today we could talk about about one Gilled mushroom in particular, and we can get into a little bit more. We can use this as a vector, to kind of dive in a little bit further into the wonderful world of Gilled mushrooms, the Basidiomycota, or some members of the Basidiomycota, I guess I should say, because some members of the Basidiomycota don't got gills. But, that's beside the point. Let's get right into it. This species that we're working with right here is a very cool one near and dear to my own heart. This is, as we can read from the tag, this is Omphalotus illudens, the eastern jack-o'-lantern. Found this guy growing on a dead stump today in good old Chesterton, Indiana. I actually had to knock on someone's door to ask if I could sample fungi from their lawn, and they very, very kindly let me, which was quite lucky because I need mushrooms for my project. But anyway, let's talk about this mushroom, and then we'll talk about some of the cool stuff, or some of the basics of Gilled fungus identification. Okay, so this is a really cool mushroom, and the reason I say it's such a cool mushroom is because this is actually a bioluminescent species. Okay, I've never seen it glow, but I've seen pictures of it glowing, so I do believe the fact that this mushroom will glow in the dark, under the right circumstances. I'm gonna try to make this one glow, but that might not work, so we'll see what happens. But you might be asking yourself, what's the function of a glowing mushroom? Well, where this guy glows is actually on the gill surface, okay? And it's been theorized, and I say theorized because I don't know if it's been proven or not. I know that there are research papers out there about this, but I haven't read them, so I don't know if it's empirically borne out or not, so I'm just saying theorized for now. But been theorized that that's actually an adaptation to facilitate the distribution of spores of this mushroom, because the gill surface will glow. The gills obviously being where the gills are hymeneal surface, if you want to get real fancy with it. They'll glow, and that glow will attract insects in, and the insects will march up and down these gills, collect a bunch of spores, and then fly around, distributing those spores for the jack-o'-lantern fungus, which is really quite badass. It's very, very similar to zookery in plants, the movement of seeds by animals. So that's quite neat. But but let's talk a little bit more broadly. Let's widen our scope a little bit and talk about some features of Gilled mushrooms. So one of the most important things that you're going to want to take note of in a Gilled mushroom is how these gills are attaching or meeting the stem. Now, there's a wide variety of vocabulary that gets utilized to describe the way that the gills run up against the stem. Words like decurrent or free or adnate immediately come to mind. And I'm not the best with my fungal vocabulary at the moment, so I can't really tell you what all those words mean. But what I can tell you is that in this mushroom, quite clearly, you can see the gills are running down the stipe a little bit here. So they're clearly attached all the way. So these are not free gills. These are, well, there's something else, Very, very, very in-depth analysis going on with the gills. So you're obviously going to be wanting to pay attention to how that gill attaches to the stem. And another thing that you can do, which is often very, very valuable to identifying really any sort of mushroom, because you can take spore prints on polypores and on toothed mushrooms. And I've got a spore print here. This is not from the Omphalotus. This is from a different species that I am trying to identify at the moment. But the way you take a spore print is I'm not going to demonstrate it because I want to keep this guy all together, because I'm being selfish and I want to preserve the aesthetic value of this wonderful fungus. But what you do is you'll lop a cap off, drop it on a piece of like white paper or black paper or tin foil, just anything that you'll be able to distinguish the color on really, really well. And you'll put that cap down and then you'll cover it up with like a cup, or I don't know, probably even like saran wrap would work. But the goal of covering it up is to keep the humidity high, because most mushrooms will only discharge their spores when the humidity is, according to what my professor told me in lecture, around like 85-90%. So you need that humidity to be kind of high for the mushroom to discharge the spores, and you want it to discharge the spores right onto the paper, or tinfoil in this situation. Because the color of the spores is often it's often quite a good diagnostic feature to utilize in the identification of fungi. So if you say you didn't know that this was Omphalotus illudens, and you were wondering, hey, what is this? You can lop off a cap, chuck it down on a nice white, black tinfoil sheet, as I've done here, and wait about like 24 hours at max typically, and then pop the cap off, and you should be, if you've got a mature specimen, you should be left with a nice spore print. And that'll at least help you out in narrowing down what you're working with exactly. So that's spore prints, that's Omphalotus illudens, and that's a little bit about Gilled mushrooms. So I hope this was illuminating to some extent. We're going to see if I can make this glow. I'll check in, I'll turn the lights all off down here, check back in around 20 minutes and see what's happening. But I think that might be the video for today. Have a good one, folks.